Okay, hi everyone. So let's continue with lecture 2 on bacterial growth and nutrition. Okay, measuring bacterial growth. So for unicellular organisms such as bacteria, so the growth can be measured in terms of two different parameters. So either it can be cell mass, where you're measuring the weight of the cell, or it can be cell numbers, like for instance CFU per mil. Okay, bacterial growth can be measured either using direct or indirect techniques. And these direct and indirect techniques can be physical or chemical measurement. For instance, direct physical measurement can be done via measuring the dry weight of bacteria, so where you dry the bacteria and then you weigh them, or it can be done via wet weight, which is including viable and total cell count. And direct chemical measurement is the measurement to measuring total protein or total DNA in a sample of bacteria. On the other hand, indirect physical measurement is performed by measuring the turbidity of your sample, which usually using a spectrophotometer. So regarding the spectrophotometer, we will learn in the next slide. And on the other hand, indirect chemical measurement is usually measuring the chemical activity of the bacteria, such as oxygen or CO2 production or consumption. And this is also directly related to the bacterial growth. Okay, indirect turbidity measurement using a spectrophotometer. So this is how a spectrophotometer will look like. And then you usually put a sample in a cuvette. It's like a small holder, and then you insert the sample into the spectrophotometer, and then you can get the reading on the screen. So spectrophotometer measures the turbidity or optical density, which is measures the amount of light absorbed by the bacterial suspension. So the degree of turbidity in the broth culture is directly related to the number of microorganisms present either in a viable or a dead cells. And it is convenient and rapid method measuring the cell growth and rate of microorganism. So let's look at this picture. So this is the blank sample, which means it doesn't contain any bacteria. So 100% light penetrated through or transmitted to the spectrophotometer. But on the other hand, when you have like a bacterial cell suspension, or let's say like a thick bacterial suspension, only partially light has been transmitted, and this indirectly measures the number of cells in the medium. So thus increasing the turbidity of the broth medium indicates the increase of the microbial mass. The amount of transmitted light through the turbid broth decreases with subsequent increase in the absorbance value. Okay, this is the summary of the bacterial growth measurement using direct and indirect methods. So maybe you can read through and then let me know like if you have any question on this. Okay, let's look at the factors that are affecting the microbial growth. As we know, microbes or microorganisms exist everywhere, including the environments in which other life forms cannot survive. And then the growth of microorganisms is influenced by variety of factors, which can be physical or biochemical factors. So when it comes to the physical factors, you can be summarized into a environmental factors, which is including pH, temperature, oxygen, moisture, osmotic pressure, and hydrostatic pressure. So let's look at the influence of pH. So usually microorganisms have an optimum pH, which they can grow the best. So optimum pH means it's the best pH a bacteria can grow. So usually if it's acidic, which is pH less than 7, or it can be neutral pH 7, or basic pH more than 7. And most microbes do not grow at pH more than 1 unit above or below their optimum pH. So usually there's a term used to classify the bacteria based on their acidity or alkalinity preference. Like for example, acidophiles. So acidophiles are bacteria that grow at acidic pH. For example, fungi, which is grow pH less than 5.5, and Helicobacter pylori, which can grow at the pH less than 3, which is in your stomach. And then neutrophils is the bacteria that can grow best in the pH 5.6 and 8, which is almost all the bacteria you're going to use it in your practicals. And also followed by alkaliphiles, which is grow best at the pH 8 to 11, which is one of the example is the Vibrio cholera, which causes a cholera. This diagram shows the pH preference of a bacteria. So the one you're going to use it in your practical mostly falls in this category. So and then this is we consider as like an ideal pH for most of the bacteria. Okay, the second factor is the temperature. Temperature is important not only for the growth but also in stopping the growth. The minimum and maximum range of temperature for different species of bacteria vary considerably, which means the preference of temperatures depends on the species of the bacteria. And then similar to the pH, the bacteria also can be classified based on their temperature preference, such as Psychrophiles, Mesophiles, and Thermophiles. Okay, let's look at Psychrophiles. So Psychrophiles are cold-loving organisms. So usually they grow best at -5 to 20 degrees, and this is including marine organisms and also bacteria that causing spot foliage in the refrigerator, which is including Bacillus, Globisporus, and Micrococcus cryophilus. And usually these bacteria are non-pathogenic to human due to the fact that the temperature they're growing is lower than our body temperature, which is 37 degrees. Okay, the next one is the Mesophiles. So Mesophiles are the bacteria that grow best at 25 to 40 degrees, and most microorganisms considers Mesophiles, that's including some of the pathogenic ones. For example, Staphylococcus aureus. Okay, the last one is the Hypothermophiles. So Hypothermophiles are organisms that love high temperature, and usually they grow best at 50 to 60 degrees. And due to the fact that they have a thermostable proteins that allow them to grow in a high temperature. And usually this bacteria can be found in the hot springs. And some bacteria that can grow at even high temperature as high as 110 degrees. And this bacteria we call as a hyperthermophiles. An example of hypothermophiles is Bacillus Thermophilus. So this is a summary. So Psychrophiles, which are like cold-loving bacteria, Mesophiles, which they love like moderate temperature, Thermophiles, a bacteria that love hot temperature, and Hypothermophiles, they love like a super hot temperature. Okay, let's look at the third factor, which is oxygen availability. So in generally, microbes have like a great diversity in regards to their gaseous requirement. So there's a few things you have to remember. So the first one is the obligate organisms, so which mean absolutely require an environmental factor for their metabolism. So in other words, it must require. Okay, and the second one is the facultative organisms are adaptable and can switch their metabolism depending on their environment. So which means facultative means they are more flexible. So it's a yes and no, but then they can still adapt. And the following one is aerobes, so which means the microorganism that require oxygen. Anaerobic means the microorganism that does not require oxygen. Okay, a microorganism can be classified into a few clusters according to their oxygen need. So let's look at obligate aerobes. So which mean they must have oxygen for their growth. Obligate mean it must, aerobe mean it need oxygen. So they must have oxygen for their growth. For example, Pseudomonas aeruginosa. Microaerophile, so micro mean it's small, aerophile means oxygen. So they need little oxygen for growth. For example, Campylobacter species. Facultative anaerobe, so facultative they're adaptable or flexible, and then they're anaerobe, so they don't need oxygen, so which came up with can grow both in the presence or absence of oxygen. For example, E. coli. Anaerobes can be further clustered into two anaerobes, which is strict or obligate anaerobe. So strict or obligate is a must, anaerobe no oxygen. So this come out with oxygen is toxic to this organism. Example, bacteroids. So aerotolerant anaerobes can tolerate oxygen but cannot survive, but they cannot use the oxygen as well. So for example is Clostridium species. Okay, this diagram shows the pattern of oxygen use. So as we know, the concentration of oxygen is usually high when it's facing the external environment or outside, and then the concentration of oxygen become low as you go into the test tube which containing a liquid. Okay, when it comes to the obligate anaerobe, usually they must have oxygen. They're concentrated on the area rich with oxygen. If it's an obligate anaerobe, they're concentrated on the region with less oxygen. If it's facultative anaerobe, usually they're quite evenly spread, but they're slightly concentrated on the area rich with oxygen. If it's aerotolerant anaerobe, so they're evenly spread throughout the test tube because they doesn't dependent on the oxygen for their growth. Okay, the following factor is the moisture. I think all of us know nothing can grow without water or moisture. So all actively metabolizing cells generally require water environment to grow. So the same thing goes for bacteria. So if there's no moisture, the bacteria can't grow. Okay, the following factor is the osmotic pressure. So the process by which water diffuses across a semi-permeable membrane from low to high concentration is called osmosis. Or in other words, the movement of water molecule from low concentration to high concentration is known as a osmosis. So most bacteria can tolerate a fairly wide range of dissolved substance by their cell wall, but drastic change of osmotic pressure can kill the cells. Okay, in the isotonic solution, usually there will be no net movement of water across the cell membrane. If it's a hypotonic related to the cell cytoplasm, the cell will gain water through osmosis. So if the medium is a hypertonic related to the cell cytoplasm, the cell will lose water by osmosis. So look at this diagram now. So if it's an isotonic solution, there's no net movement of water, which means the amount of water going into the cell and coming out of the cell is similar. If it's a hypotonic, more water will be going into the cell, which makes the cell will burst and then die. If it's a hypertonic, more water will be secreted out from the cell, which make the cell dehydrated and die. So bacteria can also group based on their osmotic pressure adaptability. Organisms that live in high sugar media we call Osmophiles, and then for example is yeast. An organism that are able to live in a very dry environment we call Xerophiles. An organism that love to live in a salty condition or salt-loving organism we call Halophiles. And one of the example is Vibrio parahematolithus, which can cause gastroenteritis. And usually these bacteria are transmitted to human through the consumption of raw or undercooked clams and oysters. The last factor is the hydrostatic pressure. As you know, the deeper you go under the sea, the greater the pressure pushing on you, right? So hydrostatic pressure means the pressure that is exerted by a fluid at a given point due to the force of gravity. An organism that can grow up to like a 1220 meter we call barotolerant. If it's up to like a 1830 meter we call barophilic. If it's up to like a 2135 meter we call extreme barophilic. And then the xenophiles are the bacteria that found in the deep ocean trench. And most deep microorganisms are barotolerant, but some are barophilic, which need a high pressure for their optimal growth. So that's all for today, and then please post your question in the e-learning, and then we will discuss on Friday.